Hello everyone, my name is Layan Elfaki, and the project that I'm presenting today examines functional connectivity reductions in the default mode network of obese patients uncovered using MEG imaging. Now, to clinically define the term, an individual is defined as being obese when their body mass index exceeds 30. Obesity is a highly prevalent phenomenon, contributing to over 60 chronic diseases, rendering it the leading cause of premature death around the world. Now, various factors contribute to obesity, including the environment through obesogenic diets and sedentary lifestyles, as well as genetic predisposition. However, given the neuroinflammatory response that results from obesity, which in turn gives rise to neurodegeneration, studies have shifted the focus to cognitive impairments that into obesity. So this includes impairments to working memory, learning, and decision-making. Now, not coincidentally, these functions are all facilitated by an intrinsic brain network known as the default mode network, or the DMN. Now, the DMN consists of frontal, temporal, and parietal regions, which exhibit high functional connectivity at resting state. So in other words, activity in these regions tends to be temporally synchronized over time. Previous studies reported the implication of the DMN in internally directed tasks, such as introspection and self-referential thinking. Now, not only is DMN activity abnormal in obesity, but it's also been found to be anti-correlated with task-positive network activity. Together, these findings support the role of the DMN as a network that actively and deliberately engages or disengages during different tasks. So putting it all together, we hypothesize reduced functional connectivity in the DMN of obese individuals. Now, as for the experimental method, 24 controls and 6 obese individuals were matched based on age and sex. So they were recruited to the Sick Kids Team Obesity Management Program clinic at Sick Kids Hospital. They were then subjected to the MEG scanner, where resting state network recordings were taken. So this was done with respect to 90 brain regions identified using the AAL, or Automated Anatomical Labeling Atlas. Additionally, because the MEG helmet records at a distance from the skull, additional statistical analyses had to be done. This was done to first eliminate noise in the data due to physiological artifacts, but also to differentiate the recorded activity to their emitting brain regions. So once done, the synchrony of pairwise cortical oscillations was identified with respect to 7 oscillatory brain frequencies, as shown here. So as for the results, the Network-Based Statistics Analysis Toolbox, or NBS, was employed to identify significant network differences between the groups. So shown here are the functional connectivity patterns that were significantly reduced in the obese group as compared to the control group. And this was done with respect to the alpha frequency on the left and the beta frequency on the right. Now, note that the low gamma frequency also showed significant results, but the figures were not available for display. Nonetheless, I want to draw your attention to the regions indicated here in red, which pertain to the DMN. As expected, this includes frontal, parietal, and temporal regions, as well as the precuneus. Now, the NBS, or Network-Based Statistics Analysis Toolbox, has been useful because it enables identification of significant networks at the global level, at the network level. However, more precise statistical corrections were then necessary to examine the significance of those functional connectivity patterns at the level of individual connections independent of network membership. And to do that, false discovery rate correction, or FDR, was subsequently applied to the data. So similar to the previous slide, the functional connectivity patterns shown here were those which were significantly reduced or impaired in the obesity group as compared to the control group. And as we can see here, for the alpha, the beta, and the low gamma frequency, not only was activity within the DMN impaired, but also connectivity between the DMN and external networks. This included the visual, the sensory motor, and the salience network. I also want to draw your attention here to the posterior cingulate cortex, which is circled in red, which was significantly affected in both the alpha and the beta frequency. Lastly, node degree analysis was computed to identify affected brain regions with the highest degree of within and across network interactions. And what we found was that regions pertaining to the DMN were among those highly implicated. But more significantly, I want to draw your attention to the fact that in both the alpha and the beta frequency, the posterior cingulate cortex, or the PCC, was the region with the highest node degree, and the significance of that will be discussed shortly. So in summary, we found that obesity resulted in functional connectivity impairments that spanned networks, and this included the DMN, the visual, the salient, and the sensory motor network. We also observed these significantly impaired functional connectivity patterns in the alpha, the beta, and the gamma bands. And this aligns with findings of previous studies implicating these oscillatory frequencies with internally directed tasks. Lastly, the FDR analyses depicting the intra- and inter-network impairments also supported the view of the DMN as a hub of transient functional interactions. So, this hub is connected to various other brain networks and thus serves to integrate information of different modalities to enable scene construction. And in turn, this enables the execution of those internally directed tasks. And a key player in this hub is the posterior cingulate cortex, or the PCC, which also plays into that integrative role as observed through the node degree analyses, FDR analyses, as well as findings from previous studies. The significance of this research emerges when we consider the grave health risks that obesity contributes to, as well as the lack of integrative research in this topic. So if we identify a brain region that's underactive in obesity, but also one that's implicated with the symptoms observed, then perhaps stimulation of that network can help not only ameliorate the symptoms, but also reduce the prevalence of obesity. So this could be done via brain stimulation techniques, such as transcranial magnetic stimulation, TMS, or deep brain stimulation, DBS. Now, one limitation of this study is the fact that many factors contribute to the development of obesity. As mentioned earlier, this includes the diets, lifestyles that lack movement, genetics, as well as high levels of circulating triglycerides. And future studies would benefit from teasing apart the influence of these controlling factors through a controlled paradigm. So for example, one sample would report poor eating habits and another would report a sedentary lifestyle and not vice versa, and thus their functional connectivity patterns would be compared to an obesogenic cohort such as the one employed in this study. Additionally, given the correlational nature of the relationship between the DMN activity and the symptomology observed, we can't infer any causality. And to overcome this, the brain imaging data could be supplemented with behavioral data to further support or validate the findings of this study. Lastly, it would be interesting to look at whether the extent of DMN underconnectivity actually correlated with behavioral measures. This includes behavioral measures of intelligence, introspection, and other cognitive functions associated with the default mode network. Here are the references, and thank you for listening.